name is Oliver Fien, and on the line as co-organizer is Dan Beard. We welcome you. This webinar is hosted by PAMNet, which is the NSF consortium of plant, animal, and microbial metabolomicists, reaching out and building a community in the United States, as well as the Metabolomics Society, which is an international organization fostering metabolomics work. In our series on grand challenges in metabolomics, today we have two speakers talking about dissecting the temporal and spatial distribution of metabolites. John McLean from Vanderbilt University, as well as Pieter Dorrestein, UC San Diego. Both speakers will speak one after the other for about 20 minutes. Following, we will have a question and answer section. If you have questions, please type those questions into your webinar control panel. So we will then collect those questions, and when the time comes for the Q&A section, we will unmute you so that you can explain and elaborate on your question, but at least we will see what kind of questions have been raised. Also, we will have a poll about your view of temporal and spatial distributions and importance of different parts. So we will also again view your the community response to this challenge. And we will try to limit it really today to 1 hour maximum, but we have not. We do not have to have a very sharp cut, but we try to limit our today's webinar to a total of 1 hour. Okay, so without further ado, I would like to introduce John McLean, who is a professor at Vanderbilt University, and I would like to give the microphone now to John.
Thank you, Oliver. And so I'd like to start by saying it's a pleasure to be able to talk with you today about what I think will be a very thought-provoking discussion, a grand challenge in metabolomics. And so it's also a pleasure being able to present with Pieter Dorrestein, friend and colleague, who will discuss spatial distributions, predominantly from imaging mass spectrometry. But to begin the discussion, we'll sort of take it in order, and so we will begin with a discussion of temporal measurements of metabolites. And what I would like to do is just to give a few slides of perspective, which can sort of be thought-provoking also for discussions about the grand challenges. So to start with, to give a sense of the way that we sort of view philosophically temporal measurements in metabolomics, we really need to break it out into two sort of disparate ideas. One is, what is the ultimate aim of the analysis? And that is to say, is it a targeted or is it an untargeted sort of discovery mode type of analysis? And to characterize those, we think of targeted analyses as usually being done, for example, using a triple quad, where you might do MRM type analyses. And there you sort of have to have the bird in hand. You have to know which of the metabolite species you're after. And so it usually provides very good quantitation and high sensitivity, but by virtue of the analysis strategy, you're limited to maybe tens or hundreds of metabolites. By and large, a lot of the research that we do more in an untargeted discovery mode is usually done using things like quadrupole TOFs or quadrupole ion mobility TOFs, usually with some amount of selectivity up front, for example, with liquid chromatography. So in those types of analyses, what we're doing is we're giving up some amount of sensitivity so that we can get broad-scale selectivity and try to characterize all of the different features that are there. Now, what I'll do is focus the end of the discussion more on that type of analysis, but specifically on how do we filter through the information to discern which are the important metabolites for the question at hand. The other way to think about temporal measurements is shown on the bottom, and that is static analyses as time points versus dynamic analyses. And each have their own advantages and limitations. So when I say static analysis, what I mean are, for example, collecting fractions at specific time points, or the time points could correspond to, say, a healthy versus a disease state. Dynamic analyses, on the other hand, is really aiming to do online metabolite and metabolomics analysis. And so there are several challenges, for example, in biological systems, removing salts from the analysis in line and so forth. And we'll go a little deeper into both of those aspects in this short presentation. So now to give a sense of what a lot of workflows look like for static time points, I'm showing some data here for a human lung cell line, A549 cells, which have been treated with a protein toxin. And so, for example, you might examine it at different time points like 2 and 24 hours. You might have different exposure concentrations and so forth. We'll do the analysis using, say, in this case, ion mobility with tandem mass spec. But then one of the problems in this analysis then is, first of all, when do we do the statistical analysis of features? And when I say features, I mean just raw peaks versus the identification. And when do we do the statistics on that? And so using nomenclature similar to that described by Lloyd, Sumner, and others, we utilize a class of identification ranging from level 5 to level 1. And as we go further and further down that funnel, we get a better sense of the validity and statistical significance of the metabolite we've identified. Now, using these static approaches, you can do some amount of statistical analysis. For example, measuring the number of unique metabolites as a function of exposure period, where you expect trends like what you see in front of you, where the number of unique metabolites increases as a function of the sampling frequency, not necessarily because there's a lot of changes in the underlying biology, but simply because you're making a higher number of sample measurements, which is one of the issues related to static measurements, and that is you're usually limited to a smaller n value, and so the number of unique things would increase as you increase n. Now this gives a sense of the data set, for example, for these lung cells which are exposed to the protein toxin. And it shows you another way to project the data, number of unique metabolites as a function of the fold difference seen under the different time point conditions. And so, for example, you can see that by and large the preponderance of species do not change very much. But what this can do is it can direct towards those species which have large fold changes and therefore are largely perturbed under the different time condition. Now, I'd like to turn to the untargeted and discovery mode analyses. Excuse me. And this is largely motivated from like a systems-wide snapshot of what's happening biologically. A lot of systems-wide measurements, I think, find their underpinnings in systems ecology. And so, for example, in the 1800s, the Hudson Bay Company kept exquisite records about the number of snowshoe rabbit and lynx furs. And what you can see on the bottom are the phase relationship of the numbers of these animals in Canada. And so as the snowshoe rabbit population increases, lagging behind it then as the lynx population explodes because there's more of a food source. So a lot of these predator-prey relationships show this phase relationship between the predator and prey. And what we're oftentimes trying to do in these untargeted discovery mode temporal analyses is to unravel the phase relationship with which different metabolites are expressed. Now, the informatics then is the problem with untargeted analyses. And so I use this slide as motivation for how we're approaching it. And in the middle, you can see it says, the Cartographers Guild struck a map of the empire whose size was that of the empire and which coincided point for point with it. Now, what this quote is really saying is it's saying that having a map which has a scale bar of 1 = 1 is really pretty useless. And so the quote goes on to say that people lived under the map and so forth. And so what we're trying to do is we're trying to find as many features as we can in the data sets, but then we need to use the informatics to rapidly funnel down to what were the interesting species, and then subsequently to do the identification once we know what those are, rather than doing all of the identification at the first step when we have the very large data sets. And that is partly motivated as well by sort of where we are in scientific history. And I would say that we live in a period where we have an unprecedented capacity to generate data. So for example, if you were to generate, say, 1 LC trace or chromatogram over 15 minutes, if you then combine it with a triple quad, you might be at around 10,000 spectra collected over those 15 minutes. Now if you do gas phase separations with things like mobility, you're up to about 100,000 spectra. And you combine that with, say, a time-of-flight analyzer, and now you're at around 10 million spectra. And I think what's interesting here is that because the speed of the separations continue to increase, this is actually underrepresenting the amount of spectra we actually collect, because we usually do MS combined with the TOF analyzer, and so we're up to around 40 or 50 million spectra every 15 minutes. So that's where the informatics plays a pivotal role. Now, to give you a case study, we do a lot of work related to the so-called human on a chip projects. So we're a part of a number of large-scale projects to emulate 3D organotypic structures on chip, which have the same function as their corresponding human organ. So there are a lot of motivations for that in pharma and other realms, and also in toxicology. And so what these devices look like is they're sort of plug-and-play systems for each of the human organs. And so, you know, I'm showing a number of those for one of our projects here for like brain and lung and heart and kidney and so on. Now, to give you a sense of the types of temporal data and the challenges associated with it, is we have to very rapidly look at what is the organ secreting into the background media as a proxy of its organ health. And so if we, for example, ping an organ with a drug or a toxin, what does that mean to the organ both acutely and chronically as a function of dose? And so this is some data for an early generation liver on chip, where what we do is we perfuse a region with hollow fibers to do gas distribution and the cell media flow. And then we also have hollow fibers which basically collect all of the waste, and that is everything the liver secretes into the background. So we seed the reactor with human cadaver cells, and we can keep these organs alive for about 2 or 3 weeks at this state. And so the workflow that we use is illustrated here. And so in this particular case, what we're doing is we're probing the toxicology of the acetaminophen exposure, or APAP. Now, ASA is acetyl salicylic acid. So salicylic acid has negative consequences on the kidneys. So in the liver context, we're using it as a negative control. And then with the kidney construct, we use APAP as a negative control. So oftentimes we're doing both static and dynamic measurements. And so step 1 and 2 is collecting fractions from the waste channel, collect the broad scale metabolomics data, and then the 2 roads divide. We can do some quick supervised statistical analysis, PCA and the like. But oftentimes what we're doing in these discovery workflows are using what's shown on step 3 as self-organizing map, or SOM analysis. Now the way these self-organizing maps work is we're utilizing them to tell us what are the important features in the data. So similar to how it's done in big data for internet commerce, this slide sort of shows you what the strategy is. So for example, when Amazon gives book recommendations, what they'll do is they'll monitor your page overs, your different book buying history, and they will create a pattern, and so sort of a temporal sequence of the data, which is shown in the map initialization tiles. Then what they do is they say that we have 200 million customers, so we have 200 million tiles, and they will create a game which is not too unlike the little kids game where you slide little tile pieces to recreate a picture. In this case, what they do is they slide the tiles so that you find which tiles have the closest correlation. And so then when they make a recommendation, they take two adjacent neighboring tiles and they tell each of those customers to do what the other customer did last. Now the way that we use it is on the trained map part on the right side of the slide, where those different pixels that are red, blue, and purple represent things like acute response, chronic response, or homeostasis. So now to make the temporal measurement, what we're doing now is sampling every minute and creating these organized maps as a function of time. So now the top row represents all of those molecules that are overexpressed upon acetaminophen exposure, and B illustrates all of those molecules which are decreased in abundance. So very rapidly what that does is it filters us from about 30,000 or 50,000 features or peaks down to only those peaks that represent the important information to understanding the prevailing biology. And so those are represented by these regions of interest, which are numbered within white boxes. So those guide us in terms of which of those molecules we need to identify. Everything else is sort of background biological noise. Now this slide illustrates a few of the different types of molecules that we find in those regions. But we find things like bile acid production begins dysregulation within about 60 seconds. We see things like drugs, small molecule conjugates, and a large number of other types of molecules. But in this discovery-driven approach, what it does is it tells us which are those other molecules that are guilty by association that might not be known biologically at the present, but when we find those clustered around things like dysregulated molecules, we can find what are the secondary and tertiary effects in the health impact of the drug or toxin. And so just to sort of wrap up, what I'd like to do is just remind you that in temporal analyses, it really is driven by the aim of the experiment, and that is whether or not you're doing targeted or untargeted really directs both the instrumentation and what you hope to achieve. In terms of temporal measurements, it also directly depends on whether or not you have the ability to do dynamic analyses almost in real time, or whether you're doing static analyses. But I think sort of the point of contention, which is really sort of, from my perspective, a pinch point in the field, is really understanding the best way to do biostatistics and bioinformatics. And now with that, I'd like to thank my colleagues that have performed the research, several of our collaborators, and these funding sources for their support. And now I would like to turn the presentation over to Pieter Dorrestein to tell you a little about spatial measurements.
Thank you very much, John. That was a very informative and very clear presentation. Again, I would like to remind the audience that you can ask questions by putting those questions into your control panel. And then after Pieter Dorrestein's presentation, we do a poll and we then answer these questions or discuss these questions. So please, Pieter. Pieter? Hello, Dr. Dorrestein?
Pieter, you're muted.
Oh, all right, now I'm unmuted. Sorry. First of all, Oliver and Dan, thanks for organizing this particular presentation. There are two things that John really highlighted that was really nice, and that is, the ability to visualize the data really leads to hone in on the specific signals that you might want to go after. Because I think in the end, if you do, let's say, an untargeted metabolomics experiment, to uncover what that chemistry is that is different is actually quite a heroic task, and I think everybody knows this, and I think we had a discussion with it on this in the last webinar that was organized here. And so what I'm going to highlight is sort of imaging mass spectrometry or spatial profiling mass spectrometry and some of the challenges that are associated with it. I do want to give credit for somebody I admire quite a bit, and that is really Rich Caprioli. I think, to me, this is the article that placed imaging mass spectrometry on the map as a routine tool. There are some other people that have done spatial profiling before, but I really think that this is the key paper that drove this. And what he did in this particular paper was that essentially he had some cells or some ink that he had deposited, and then he subjected this to imaging mass spectrometry, meaning that he simply rastered across the sample with his laser. In this case, it was a MALDI-based system. And at each one of these pixels, you collect an individual mass spectrum. And so what happens is if you have two or more of these pixels that contain that same ion that you might be interested in, you start to see a distribution associated with this. And so here he has a Kamasi stain C with a circle on it, and he simply rastered across all these different points and then colored the specific ion that corresponded to Kamasi. And then he showed this with a biological sample where this is a fluorescent image of some human-derived saliva cells, essentially, and then here's the artificial stain for a protein with the mass of around 7,000. But this really showed that you can begin to do the spatial profiling with biological systems, and in fact, you can apply this to almost any biological systems these days. And so there's a lot of choices you have to make when you start if you want to do imaging mass spectrometry. The first one is, do you want to apply matrix, or do you do that without matrix, so matrix-free or a matrix application? And I'm going to go through each one of these steps throughout this process. But essentially, you have your sample. You then raster across your sample, and there's two ways of doing this. There's either the continuous rastering, and then to create a pixel, you then time bin and average that spectra in that particular time bin to create your pixel, or you jump from one place to another place, and then the next place, and then the next place, and you collect individual spectra. And so if specific ions, if you're in one of these pixels and you see a specific ion, you now can color that specific ion in this spatial profile. And so here we would have an M over Z 1, 2 versus 3, and you get different spatial distribution. Then there's a step of data processing, which a lot of people skip over, but I think it is actually quite important, and that involves, let's say, denoising, for example. And then you merge your signals, and that begins to get you to some interpretation or hypothesis generation in terms of what signals are important. And then you need to do compound identification, and it leads to predictive molecular models in biology and medicine. And ultimately, I'm going to address data storage and accessibility. So in this first step, in terms of matrix application choices, there's actually a lot of matrices that you can apply. And typically, my advice when somebody says, I'd like to know what matrix to use for my specific application, there's some bias that you can do. So if you are looking at peptides and proteins, dihydroxybenzoic acid and synapinic acid tend to work very well. But my advice is ignore the literature and do it by trial and error in many ways. Purity, I think, of the matrices is an important consideration, as purity affects crystallization. We have a really hard time in imaging mass spectrometry, particularly when you apply matrix, to reproduce some of the results. And I think part of it, even when you use the same matrix, if the purity is very different, or your solvents that you're using to promote crystallization is different, you might get slightly different crystal forms and size of crystals, and that affect what you're going to see. And there's a lot of different strategies also to apply matrix, and these are just a couple of them. There's wet methods and dry methods. The wet methods that are commonly used are essentially a paintbrush. So this is that you can use for model kits. You put your matrix in there and you literally spray it on top of it. These were some of the earliest images that were created with this particular approach. There's an acoustic pulse where you essentially hit pulses of droplets. What's nice there is that you can, let's say, if you're interested in peptides, you can first apply trypsin, so you can digest your proteins and such, and then apply a second spot right over that same place where you put trypsin on in terms of application of matrix, etc. And then there's a variety of devices that are out there where people have developed methods to apply matrix. This is one of the ones that's sold by Bruker, but there's a variety of other ones that have been commercialized that are available now. We don't have a really good handle of when should we use which one, and what I found out that it's usually person specific. Some people are very good with this manual spray, and some people tend to work only with the acoustic spray, and some people with these commercial applications. There's a series of dry methods. The two most common ones are the sublimation approach, where you sublime the matrix directly onto your molecules or onto your samples. And that's usually applicable for smaller molecules. I've not seen too many applications of sublimation when they wanted to look at proteins, for example. There's also dry coating. This is something I use very heavily in my laboratory, and I'll show you an example in a second on how we use this microbial imaging mass spectrometry. And then there's some matrix-free methods, and the NIMS plate that was forwarded by the Shoestack lab is one of such examples where you essentially have a coating of or clathrates, essentially the holes in your matrix plate. And those are filled with a liquid, and on top of it, sometimes you have a thin Teflon layer, for example. And then when you hit it with the laser, you essentially create a mini explosion that then, if you lay your sample on top of it, then it creates ions in the gas phase. And this has been very useful. The one limitation is to make these NIMS plates has been fairly challenging. And it hasn't been commercialized also, although I hear through the grapevines there are some people beginning to commercialize these particular tools. But in my case, what I use the dry coating for is microbial imaging mass spectrometry. We grow microbes directly on Petri dishes. We cut out the agar. We move the agar to a multi-target plate shown right here. And then once it's on the multi-target plate, we then cover the sample with a matrix, and in our case we use a 50-50% mixture of synapinic acid and dihydroxybenzoic acid, and we use this for both positive and negative mode. And this is why I say to a certain degree you should ignore what's in the literature and actually test on your samples whether the matrix can be used for the applications that you're interested in. And then we cover the sample with a matrix, and in our case this is possible because the sample itself is wet. So when you sprinkle the matrix on top of a wet surface, it dissolves a little bit, and then it recrystallizes, and you get a uniform distribution. And so this has been working really well and it's a really easy method for microbial imaging mass spectrometry. And then once the sample is covered with the matrix, you then subject this to imaging mass spectrometry. And here I show a multi-base, so you hit it with the laser, but you could also do DESI or nano-DESI type approaches. But you rest across your sample. You collect individual spectra at each one of these locations. And then if you're interested in a specific ion, so let's say this green ion, if that's observed in that pixel, you would color that green. If it's a neighboring pixel has that same ion in there, you would color that green as well. And what it does, it creates an image because you will have multiple pixels. So it's very similar to the images that's seen on your computer screen. That your computer screen is actually generating a lot of different pixels, and if those pixels have the same colors, you will create an image. So the only difference between a computer screen and imaging mass spec is that it contains molecular information. And so there are a variety of different ion sources that are available to do imaging mass spectrometry, but I think the three most popular ones that are most commonly and widely used are MALDI, matrix-assisted laser desorption, and that relies on your sample and then covering usually with a matrix. In some cases, you can eliminate the matrix, but that's usually only reserved if you have some sort of chromophore in your sample to desorb the ions and get them into the gas phase. There's secondary ion mass spectrometry. One way this works is that you hit it with a sample of buckyballs. You have your analyte, you accelerate the ions into the gas phase. This is a matrix-free application, and there's some really nice biology that has come out of it. The other approaches are DESI or nanodesi, but DESI, the way this works is essentially you have a solvent flow that hits your sample, and it bounces up molecules or droplets into the air, and then you basically capture those droplets with the mass spectrometer, and you can use raster over a sample and get this kind of information. And of course, there's lots of advantages and disadvantages between all the methods, and I always think of these are all complementary methods. They have a different mass range that you would utilize these for. They have different resolutions that you can scan them with. They have different probe depths, and of course, there are different costs associated with each of these technologies. And so when you start an imaging mass spectrometry experiment, you need to think about, okay, what is my question? What are the key parameters that I need to know? Do I need to have intact molecules, or is it okay if I break apart my molecules? And so those are decisions you're going to have to make. And then once you collect the mass spectrometry data by either three of these methods or many of the other methods that exist, and I didn't highlight comprehensively, there's so many different ion sources that are out there, and you can use almost any ion source for imaging mass spectrometry. But once you collect your data, you now get onto creating the molecular image. And there's really a variety of different approaches, but there's data processing, and then you have to merge your signals. And here are some representative examples of MALDI visualization tools. What is notable is that many of them are not freely available. Most of them are actually you have to purchase. But I think this is going to change over the next few years. There's going to be many, many more tools that are going to be freely available. Biomap, of course, has been around for a long time now. Bruker is one of the premier tools, the flex imaging software. But recently, Skills has come on the market, which is one of the only ones that can also do three-dimensional reconstruction, irrespective of whether you're doing DESI, nanodesi, MALDI. You can build three-dimensional reconstructions. The one question that we have often is, okay, what software might be better for specific applications? Right now, there's no guidelines for this, so we don't know which one is better or for specific applications, and that needs to be investigated at some point. Imaging mass spectrometry can be done in the third dimension, and here's a review by the Caprioli lab on this particular thing. But the way imaging mass spectrometry, the way you have to think about it is, okay, I can take an image from the top. So this is, in this case, I'm looking at a microbial colony, but you can think of this as an animal or a plant. But there's always a third dimension associated with it. So here we're looking at some features of this microbial colony underneath in the agar environment where this microbial colony is growing. And so one of the things that you can do is you can simply section this particular sample that you have, and then subject these sections to imaging mass spectrometry so that you can start to see the chemistry that is associated with these different features that you see. And so similar to what John was talking about, we need to focus in on specific molecules that might be of interest. So here we have a molecule with a mass of 522.5. It's only seen in the tips of this microbial colony. So these are the root-like features that are associated with this microbial colony. And so if you think of this microbe maybe sitting on a skin tissue, maybe these type of molecules are involved in the lysis of human cell during an infection. So that would be a hypothesis, and then you start to focus and spend a significant amount of time and effort to figure out what that type of molecule is. And so you can see a wide range of distributions, but you can also think about, okay, there's 3 dimensions to this particular colony. So if I take multiple slices, I can then begin to see what happens in 3 dimensions if I can reconstruct this. And so that's shown, here's an example of this, where we see all the chemistry that are associated with 2 microbes that are interacting in terms of the chemistry that's underneath in the agar environment. Here, the Cook's lab has done this very nicely with a 3-dimensional model of the human brain, which was also the sample that Caprioli used in 2008 when they first forwarded 3-dimensional microbial imaging mass, or 3-dimensional imaging mass spectrometry. Compound identification, I think this is truly the bottleneck in mass spectrometry. So let's say you're interested in specific signal. You really need to take out the kitchen sink to try to figure out what that molecule is. And in some cases, this may be quick if you have reference spectra, let's say MSMS spectra in databases. But if it's not present in the databases, you need to isolate it and do the structural elucidation. That is often person years of effort that goes into that. And we discussed this a lot in the last Grand Challenge webinar that we had. Then I want to get to the data storage and accessibility. So if I look at how imaging mass spectrometry is used or reused, the only people that sometimes reuse imaging mass spec data is when they do new computational developments. There's nobody that I know of that has actually uncovered new biological insights after the initial paper that was published. And part of this is because the data that is generated by imaging mass spectrometry is simply not accessible to people who want to look at it. Let's say neurobiologists. They may want to look at the 3D brain map. This is simply largely inaccessible. And the imaging mass spectrometry community has been thinking about this. I'm part of this paper to GigaScience, where we provided benchmark data sets for 3D MALDI and DESI imaging mass spectrometry. Ben Bowen is developing an open resource, an open MSI resource, where anybody can go in and view different imaging mass spectrometry data sets. But and these are the right developments, and this is good for the community. The limitation that Ben still has is the volume that he can take. So, for example, when I asked him, can we upload 1,000 of our imaging mass spectrometry data sets, it was not yet possible. But on the other hand, having accessible analysis infrastructure for the community, I think, will become very valuable. And I think Ben Bowen is certainly leading that particular effort. And so imaging mass spectrometry is one way to do this. You can also do spatial profiling. The idea is actually very simple. You can have any 3-dimensional object. You can spatially collect samples and then use that data to provide some information in terms of the spatial distribution and build hypotheses in terms of biological function. And so in this case, what we can do is we can take samples, and in each one of these different locations, we can subject them to either MALDI-TOF or UPLC QTOF analysis, and then we can take the intensity of the MALDI-TOF data and map it back, or take the area under the curve in the UPLC QTOF data and map it back onto these 3-dimensional models. And this is really the type of information that you see. So in the upper right-hand corner, this is a MALDI-TOF data set. You see the M/Z value, and then blue is low intensity, red is high intensity. And all it's doing is, as you're scanning the M/Z range, you start to see that there are different distributions on this particular person. And so I'm just going to highlight one example of a distribution that we saw. and that was this particular one. It has a mass of 3372. The only place you see it, there's a red dot right in the neck of this particular female. And I'm going to zoom in on this. If we actually look at photographs that we took during the sample, what you will see is that there's a big red pimple there, and that pimple showed very specific signatures. One of those was a molecule with a mass of 3372. It's called the human neutrophil defensin. So this is typically a molecule that's upregulated when there's an infection taking place, and that showed up very nicely in this particular image. And so you can extend this to many other different things. But what's nice is that you can begin to incorporate many different analyses. So here we have an example of lungs that we're investigating, and we can map microbes, which are on the top, and the molecules that actually come from these particular microbes. And so you can clearly see that some of these molecules that come from these microbes are only seen in the top and not the bottom. And that shows that there's very different microenvironments that exist within these particular lungs. But you can, in principle, do this with any other system. So here we're looking at a plant. Here we see a microbial molecule called palinomycin that covers the entire plant surface. Here's my office that we've been able to map. And so here are 4 people that were sitting in my office. And what you will see is that one person takes Norvasc for high blood pressure. You see another person that utilizes avobenzone. But the key message to take home is that visualization really drives rapid interpretation. So if you look at this particular office image that's rotating, you can see a signal on 4 people, and you can see a little bit on the floor there. Now, if you look at the one person, happens to be me, I don't have any of that signal on me. And it turns out that the signal is caffeine, and I don't drink caffeine, but the other 2, particularly the 2 males that are sitting adjacent to me, they drink a lot of coffee. And then I figured out, well, somebody spilled coffee in my office because we can see it on the floor. And so very quickly we're able to interpret what might be happening in this particular context. And you can apply it to office studies, you can apply it to medical studies, you can apply it to so many other different things. So some of the grand challenges that we didn't discuss is really how do we translate spatial information to gain biological understanding? The annotation of signals. There's a lot of imaging mass spectrometry. We publish it. There's a couple of signals that are identified, but there's no curated place that we can go to say, okay, if I see the 3375, it is this particular molecule because it's been seen in these particular imaging mass spectrometry data sets. What that would do is actually it would help you to figure out what you need to elucidate if it becomes an interesting signal and what you should not spend the time on. We need to be able to compare and contrast data sets, and for this, data accessibility is critical. It would be great at some point that we can take study A from person A, similar to the way genome sequencing is done, that we can grab a genome and compare it to my genome that I have. I think these possibilities exist within imaging mass spectrometry as well. An easy integration of, with other imaging modalities. There's an example here with Graham Cooks and Natalie Auger, where they integrated sort of MRI data with mass spec profiling. If we make this easier to do and accessible for everybody, I think it would really provide, mass spec will become integrated into many other platforms. We really need to think about how we convey statistical tools for time and spatial information. I think SOMs, such as the one John was talking about, it was very useful to begin inform what are the most relevant molecules that might exist. And SOM is one example of a statistical tool. And then finally, I want to thank friends and co-workers with 3 people in particular, the Bandera Lab, who we do a lot of chemical identifications with, the Alexandrov Lab. All the 3D mapping that you showed was really driven by him, formerly in my laboratory, but now independently at EMBL, and then Night Lab for sequencing. And then I will take any questions from here.
Thank you so much, Pieter. That was truly fascinating, I think, for all of us to see that. So while we collect questions, and I see that there's not many questions yet, Dan, can we maybe just do the poll first?
Sure, I'd be glad to coordinate that. So this is a series of 10 poll questions that John and Peter thought would help them and to help the community in terms of this type of technology. So your screen will go to just a question, and we'll take a moment for you, everyone to analyze, to choose to answer it or not. So first question, do the majority of your metabolomics experiments utilize a spatially resolved sampling protocol? So please answer yes or no. Okay, we've got about 70% participation. All right, we'll go to the next question. The next question is, do the majority of your metabolomics experiments... I'm sorry?
Where can we see the results?
We'll do that at the end.
Okay, sounds great. Thanks.
Do the majority of your metabolomics experiments utilize a temporally resolved sampling protocol? Okay, about 70% participation. We'll go a few more seconds. 3, 2, 1, done. All right. Next question is, what is your most common temporal sampling time frame? And you can select just one of these. Okay, about 50% participation. All right, we'll let this one go in 3, 2, 1. Next question. Do you utilize any of the following in your metabolite workflows? And you can check all that apply. All right. Almost 50% participation. Okay. Yeah, this is a complicated question, so it's coming up. 65% participation. All right. We'll stop this question in 3, 2, 1. Next question. Do you perform metabolite IDs following or before statistical analysis of time series data? So those of you who are doing time series data. Okay, that's a little over 50% of participation. Okay, we'll stop this one in 3, 2, 1. Next question. Do you use MSI or spatial profiling in your metabolomics workflows? Okay, we're almost to 70% participation. Anyone else? Okay, we'll let this question go in 3, 2, 1. Okay. What MSI method have you used? Select one of the following. Everybody should be able to participate in this one. 60%, 64% have voted. Let's get to 70. There we go, 70% participation. All right, this one will go away in 3, 2, 1. Okay. Have you deposited an MSI data set and made it accessible to the public? And if you have, there are some choices there to see which mode you've used. Okay, we're over almost 60% participation. Right at 60% participation. Anyone else? We're going to shut this question in 3, 2, 1. Next question. Do you perform matrix-free or matrix-based MSI? Or both? And everybody should be able to participate in this one. Okay, we're almost at 70% participation. We'll close this in 3, 2, 1. And the final question, have you integrated multivariate statistics such as PCA or segmentation into your MSI data? Okay, we're about 50% participation at 30 seconds. And we will close this in 3, 2, 1.
All right. Thank you so much, Dan, for this poll, and I understand we will see the results very soon.
Yeah, we can go to them now if you want.
Sure, why not?
And Peter and John, if you would chime in on your impressions of these. So this is the first question. So spatial resolved sampling protocols. I guess a clear majority do not do any spatial resolved sampling.
Yeah, I don't think there needs to be, this one doesn't need any chiming in, I don't think.
Okay, all right.
That's great.
Temporal sampling. This one's more equivalent. About half of the people do. Half of the respondents do.
Mm-hmm.
Yeah, so this is John. What I would suggest is that this also represents that sort of dichotomy of static versus dynamic, because many static measurements could be interpreted this way.
Okay. What is your most common temporal sampling time frame? Looks like to me it's mostly on the scale of days. But a few people are doing sampling on the second time scale. All right, no comments. All right. Do you use any of the following in your workflow? Metabolite workflow.
So I guess, so this is John again. I guess one of the interesting one is the derivatization. So I'm probably guessing that most of those are GC strategies. But it would be interesting for me to know more of if other analytical workflows are using derivatization.
That's a good point, and we could make that a challenge discussion in some later point.
Yeah. All right. Do you perform metabolite IDs following or before statistical analysis of time series data? Looks like the majority do that after.
Okay.
So I guess you look for statistically significant features and then try to ID them. All right, let's hide that. Then, so here it looks like a clear majority of the people are not using MSI or spatial profiling in their metabolomics workflow. Is that surprising to you, Peter? Or?
No, not at all. I think that's reflected in the community. So that makes a lot of sense.
All right.
I think it's becoming more popular, so I would have been interested if we started this poll 10 years ago, see how much it's creeping up.
Oh, yeah. Okay, the method of MSI. Looks like Maldi's the winner there in terms of, does anyone want to raise their hand and maybe say, so you can electronically raise your hand, and if you would tell us what your others are, that might be interesting. All right, I don't see any hands going up, so we'll go from that. Okay, so very few people have deposited any data anywhere, it looks like. And about, to me, it looks like it's evenly split between some of those public repositories and things like dedicated web links.
Yeah. Now, I would be interested maybe in a future poll to actually do this for metabolomics, not MSI data, and see how many people have actually made data accessible. As I highlighted last time, I could only find 558 projects in the public domain, and that's between the Metabolomics Workbench, and GMPS.
Well, it's certainly improving and changing. So.
Yeah, no, I think it's fantastic. We're on the right track.
Okay, matrix-free or matrix-based MSI. I don't know. So yes. 70% of the people aren't doing it at all. So
yeah, that's it. Yeah, but it looks like the majority are doing both, which is great. Because they really complement each other in terms of the type of information you can get. So it's fantastic.
Right. And the last question, have you integrated multivariate statistics into your MSI data analysis? And that looks like it's split. So that's the poll, or the polls. And I'm not seeing any questions. If anybody has a comment or a question, if you would raise your hand electronically.
I do see a question that's asked by Kent Chapman, whether or not slides will be archived for later access.
I think the whole presentation is on video, right?
Yeah, we should.
I think these are stored at the society website, or?
That's the intention, and Lloyd has put one at least on YouTube, so they'll be a lot more available in the next month.
Yeah. And otherwise,
I think
if they want the slides, I'm sure they can email me or McLean, John as well, and we're happy to share. At least I am.
Okay.
Yeah, I don't see a lot of discussion points here either, but I would like to say that we will continue next month with Philip Ritz-McKibben on improving metabolomics cost, throughput, and data comparability. So again, invitations will be sent by emails to the community. So the November will be on cost, throughput, and data comparability. Unless there are now people raising further questions, and I don't see anyone right now, I would like to thank the presenters very much, John McLean from Vanderbilt University and Pieter Dorrestein from UC San Diego for the superb questions they have raised and challenges they have explained. And we, I think, are all very much enthused to think about better integration of temporal and spatial resolution into our workflows, into our studies, because these obviously make a lot of differences in terms of biological interpretations. So thanks a lot for both presenters. And we can't clap your hands, but I'd say thank you so much, and yeah, see you in next month.
Thank you.
All right. So people are dropping out.
Yes.
I think this is now closed. Thank you, Dan, for organization.
All right. Thank you very much. I'm going to quit the meeting.